Hello, everybody. I'd like to welcome you to HCG Molecular's Nature webcast. My name is John Lucky. I'm Senior Director for NGS Assays here at HCG Molecular, and I'd like to thank you for joining us this morning. I'd like to introduce our speaker, Dr. Mark Stern, who will present our presentation, Utilizing Gene Expression to Integrate Biomarker Data and Illuminate Complex Biology. Mark has been with HTG for 5 years, and in the past 2 years has served as our VP and head of the Immuno-Oncology franchise. In this role, he's functioned as our corporate subject matter expert, an expert on immuno-oncology, as well as being responsible to help drive new product development, craft corporate strategy, and managing some strategic key scientific collaborations for both our KOLs and our SAB. Prior to his career with HTG, Dr. Stern spent 10 years in academia and earned his Ph.D. from UC Berkeley in molecular and cell biology. At this point, I'll hand it over to Dr. Stern.
Great. Thank you, John. And good morning to everyone. I'm pleased to have this opportunity to be able to speak to you today and to share more around our capabilities in immuno-oncology and in IO. To start out, as a general intro, it's been yet another great year for IO as a field. We've seen that the pivotal usage of immunocheckpoint inhibitors has expanded into new indications. We've seen the emergence of new combo regimens, essentially addressing new mechanisms of action, and we've seen new insights into disease biology driving essentially disease biology and immunoscape mechanisms. So as a molecular diagnostics company, as HTG, we're extremely excited to play a role in this. Just by way of a brief intro, I wanted to give those of you who are unfamiliar with HTG a brief background into both our capabilities into molecular gene profiling as well as an understanding of our chemistry. But then I really wanted to spend some time sharing some specific collaborations and specific applications with HTG in the field of immuno-oncology around two key collaborations. And then I wanted to give a brief introduction around a newly launched assay that we're very excited about with my group, our new Precision Immuno-Oncology Panel, or PIP. Great. So as a company, HTG is a molecular diagnostics company. We focus in molecular profiling. So as a company, we provide these capabilities both through a services and a product-based model. Essentially, we're really focused in terms of developing molecular tests that we hope to utilize as diagnostics to really empower local decision-making closest to the patient, during treatment. And so as a company, HTG is engaged in both producing RUO assays for retrospective screening, as well as developing prospective assays as part of existing companion diagnostic agreements with pharma partners. We're excited because as a technology, we believe that HTG can address some critical questions within the field that I'll get to in a moment. So I'm sure mostly everyone is familiar with this slide in one form or another. This is a schematic that's pretty widely used in IO. And I just simply wanted to point out more or less from a diagnostic standpoint, from a perspective that, as we look at those roughly 10 to 30% of cancer patients that are likely to see benefit from either immunotherapies or a combination, we see that getting those patients onto the tail of the curve so that they can recognize a deep, durable benefit is key. And so part of that is really identifying better predictive biomarkers that can find those patients likely to benefit from those monotherapy settings for immunocheckpoint inhibitors, or potentially to find combinations or regimens of drugs with different mechanisms of action that might particularly bring benefit to these patients. But in either case, really the clear unmet need is to really identify new biomarkers that can really help guide either the treatment of patients in a monotherapy or in a combination setting. And so this really brings us to the challenge, I think, in the field, which is that of tissue availability. So as a transcriptomics analysis platform, HCG is addressing what we believe to be a critical need within the field. So if you consider, as we've been becoming more sophisticated in our biomarker strategies, specifically in IO, as we've moved more from a all-comers essentially treatment across the population to looking at specific biomarker-selected populations, what we see is that the tissue need to be able to generate all of the relevant biomarker data is extremely costly. And as we now move into what we see as a total omics field, where we're looking at different platforms and integrating different biomarkers together into a single analysis as part of precision medicine, we see that there's a huge need at this point to be able to derive as much biologic data as we can from these clinical samples to really inform treatments. And so this is essentially where HDG actually is able to provide the most value. Essentially, as a targeted sequencing-based methodology, we can actually use molecular profiling assays together with a nuclease protection-based chemistry with NGS to essentially perform targeted sequencing in a crude lysate. So we're able to take a tissue to permeabilize it and then to measure mRNA levels. And we'll explain in a moment how we can do that. But the end result of this is a multiplexed assay with a high sensitivity and a very broad linear range of detection. So this is the EdgeSeek, which is the HDG platform that we use as part of our molecular profiling capabilities. And you can see in the schematic, really how we operate as a technology. We typically, in many cases, are going to be working with FFPE tissue. We simply excise a tumor surface area of interest or a geography that we want to look at, and then essentially permeabilize that in a proteinase K-based buffer. And then we perform a nuclease protection assay with S1 nuclease. And what this does, in a very general sense, is give us a molecular impression of the transcriptome in that tissue, essentially giving us an idea of what genes are expressed. And so we're able to utilize both PCR and NGS downstream to essentially adapt this into the NGS realm to give us a molecular profiling assay. And so this is how the chemistry works, essentially at a very general high level. Each of our assays consists of a mixture of oligonucleotides, nuclease protection probes of roughly 100 base pairs long. There's a core 50 base pair region, which is the reverse complement to the mRNA sequence that we're looking to detect. And so in essence, what we do is we perform an S1 protection reaction and then essentially take all of the probes which are protected from that enzymatic reaction, which are generally a one-to-one stoichiometric relationship, and then use PCR to essentially tag the PCR amplicons so that we can perform multiplexing as well as NGS workflow. In terms of as a company, we've developed a number of assays, a number of content that allow us to perform retrospective analysis of different clinical samples. In particular, we've developed a number of assays that have been previously released, both mRNA-based and microRNA. And several of the collaborations I share today will refer to our oncology biomarker panel, but I will also refer to our new precision immuno-oncology panel as well. So just briefly to give an overview of our new IO panel that we're very excited about, this is essentially a 1,392 gene panel that's really targeting different immune pathways as well as uniquely different intrinsic tumor biologies that we've built into this assay that I'll show on the next slide. As I described before, the technology is adept. It allows us to interrogate small tissue samples by virtue of the sensitivity of NGS that we use, as well as working with a range of samples. And very, very critically important is the fact that we have experience working across multiple different, I would say, qualities of samples, typical of most multi-site clinical trials where we see a variability across different FFPEs. And so the ability to work across all this and to address that variability is a critical aspect of our assay. This is a schematic of the content in the Precision Immuno-Oncology panel, or PIP panel. So it incorporates a number of different immune pathways as well as biomarkers that essentially have been newly found or discovered, including different checkpoint resistance pathways. And we additionally built in functionality around the TCGA, essentially to look at different molecular expression phenotypes as we're seeing this more prevalent within the field. But most importantly with assay, what we're very, very excited is the ability to provide applications, the ability to use this assay to understand critical aspects of the biology, particularly looking at immunophenotyping, which is deconvoluting some of the expression, some of the immune pathways to better understand what immune cell subsets might be present in the tumor microenvironment as a way of understanding different immunosuppressive cues or other aspects of the biology. In addition, HDG is extremely excited to focus on tumor inflammation as well and to better understand hot versus cold tumors, something that we're currently developing applications around, and several other applications that we're going to have. So overall, we're very excited because we believe that PIP is going to essentially deliver what we believe a more comprehensive analysis of the biology driving tumor pathogenesis, as well as the surrounding microenvironment and associated immunoinhibitory signals. We believe that this is going to be critical as we move or turn to the next chapter of IO, which is really deconvoluting a lot of the immuno escape mechanisms that are typically in play in most standards of care that we're seeing. So I'm very excited to share a couple of examples towards how HDG is really actively engaged in IO. And as a company, we're very invested within this field and really looking to play a bigger role within this landscape. And so the first study that I wanted to highlight is a very eloquent study done by one of our collaborators in the Center of Léon Bérard in France, where they were trying to understand molecular subtyping in head and neck cancer. So oral squamous cell carcinoma is a type of head and neck cancer. Head and neck is a very heterogeneous group of cancers, as probably many of you know. And actually, there's huge unmet medical need in this area, essentially. And so one of the questions that Pierre wanted to do is to use gene expression profiling to better help understand what aspects of the tumor biology or the neighboring biology in the tumor microenvironment might be helpful in terms of thinking novel therapeutic approaches to be able to treat these patients. And so interestingly, Pierre wanted to focus on understanding a particular group of head and neck patients, never drinker, never smokers. And it's known that these are etiologically different patients than drinker and smokers. And so one of the key questions is, what is leading some of the differences clinically between these two different patient subgroups? Paradoxically, it's known that the never drinker, never smoker, have a higher amount of inflammation in their tumor microenvironment. But interestingly, they don't have an elevated level of tumor mutation burden or other types of mutations that are otherwise different than what's seen in the drinker-smoker part of the population. And I should say as well, this excludes the HPV component as well of this cancer group. But in either case, though, Pierre is looking to understand what is different in these never drinker, never smokers by the fact that there isn't a lot of mutation that's differentiating them from the drinker-smoker cancer patients. And so Pierre started by first doing what many groups are doing, is taking advantage of the richness of the TCGA network out there and essentially interrogating this, looking for transcripts that are differentially expressed, essentially, between these two different cancer groups. And what you're seeing here is actually the initial results that he had found, which was that roughly 850 genes were found to be differentially expressed, as found by looking in the TCGA data sets. Roughly 219 genes were overexpressed. And what he was able to do is to calculate an enrichment score, to compute this, and to really validate this result in additional cohorts that he had access to for these oral squamous cell carcinoma. And you can see here on the right, two cohorts, the GSE cohorts, where you see higher levels of enrichment scores for this 850 gene set that's expressed in the never drinker, never smokers versus the drinkers and smokers. Okay. This is essentially a slide that addresses how HDG fits in to this particular study. We're very excited that Pierre, as part of his study, really was limited by how much tissue he had available to be able to discern or to study different pathways per se in his clinical sample. So he turned to HDG and the fact that we can work in many cases with a single section of FFPE to be able to generate biologic insights. And so what he was able to do is to take advantage of one of our existing products, our OBP, or Oncology Biomarker panel, which has over 2,600 different genes, and he found that roughly 120 overlapped with this panel. And so using essentially machine learning and deep learning techniques, he was able to compute an enrichment score even with this partial truncated set from OBP, and was able to show again that he can reproduce what he saw in the previous cohorts with an elevated never drinker, never smoker score relative to drinker smokers. And on the right side is really the exciting aspect of where he looked to validate this result. So he had used our technology to generate a 25 gene interferon gamma signature. So if you remember, I had said earlier that the never drinker, never smokers tend to have more inflamed tumor microenvironments, which distinguishes them from the drinker smoker patients. And what we found is when you rank order these patients in order level of interferon gamma expression, which is a sort of a surrogate giving you an idea of existing immunity that might be there, potentially anti-cancer immunity, we're seeing that as you look at increasing levels of interferon gamma, we see PD-L1 in IDO1, two markers that we know are downstream and regulated by interferon gamma signature, upregulated. This is exciting because, again, this was detected on our EdgeSeek platform. We went on to validate that this tumor inflammatory data was actually consistent with our orthogonal biomarker data that we determined by IHC. So on the bottom, the blue-red bar diagrams indicate IHCs, where in red we're looking at a high level of IHC versus a low level expression. And you can see that PD-L1 and IDO1 both were enriched and upregulated in the patients that had a high level interferon gamma signature, which is what we'd expect. And if we look in the T cell compartment and we look at both a pan T cell as well as a T helper Treg as well as an effector T cell, we see again enrichment of these different IHC signals together with what we're seeing at the transcript level. And so this slide here essentially shows you, part of the data, and I can't tell on my screen, it looks like I'm missing some of the IHC data that should be there. But in essence, what Pierre had done was he turned to his pathologists and essentially were looking to compare how good did HDG perform relative to his pathologist. And really excitingly, we found when we were able to see an enrichment of different signals is determined by immunostaining, whether that's looking at peritumor expression or looking at different immune cells expressing, let's say IDO1 or looking at PD-L1, we're able to see an upregulation of T cells that were infiltrating the tumor microenvironment, as well as the expression of other immune inhibitory receptors, suggesting that, again, we can validate these results and see, in fact, that the expression that we're seeing from EdgeSeek is, in fact, making sense, and it completely aligns to the biology that we're seeing. I wanted to, as well, when I have, with the time that I've left, really highlight another exciting study that HDG was involved in and was really excited. And this was having to do with the Checkmate 275 study with BMS. And so this was a single-arm study evaluating the safety and efficacy of nivolumab, an anti-PD-1 antibody, in a second-line setting in bladder cancer, urothelial carcinoma. This is a particularly very important setting in that this is huge. This has a huge unmet need, and as a testament to that, was given breakthrough designation and was approved under accelerated conditions. And so, in this particular study, HDG was involved in terms of studying an interferon gamma signature. And so there's a lot of complexity to this Lancet paper that came out the previous year. I encourage all of you to look at it. I won't have time to get into the really deep science of this, but I will point out that when we were able to look both essentially at the interferon gamma signature as well as the TCGA molecular subtype, we're able to see interesting correlations with the clinical outcome of the patients that were responding to nivolumab on this phase 2 study. On the bottom, what we're looking at is more or less a score. This is an interferon gamma score comparing both the signal in disease-controlled patients with patients that were progressing with their disease. And we saw a statistically significant increase in that score in the patients that were benefiting from nivolumab. Now, on the right, I did want to point out, and this is a bit of sobering news for many of us, and this is really a testament to some of the work that's being done by Oliver, Michelin, and Lucene, that, we really need to do better when we're looking at our patients in terms of response, non-response, and really finding biomarkers that optimally resolve essentially the biomarker plus and minus, really having better negative predictive values. So ultimately, this is a great start for all of us, especially since PD-L1 IHC was not found to be especially predictive on this trial, and the fact that interferon gamma worked really well off of a single slide of FFD was exciting, but we're obviously doing more to build off of that. I wanted to present a corollary or an addendum of the story, again, that was very exciting. That just recently came out in just a week or two ago from essentially the Gillespie group in collaboration with BMS and MD Anderson, which was actually addressing a critical question that we at HDG are really spending a lot of time thinking about and addressing with our different assays, which is why are some hot tumors not responding effectively to immunotherapy, particularly immunocheckpoint blockade treatment? And what we found in the study, which is extremely exciting, and again, I encourage you to look at this paper, was in fact that even for tumors that have infiltrating T cells with effector, cytolytic, functions that have been upregulated and essentially differentiated and proliferating, what we find is that signals from the stromal compartment or from a stromal tumor interface are directly influencing the activity of the PD-L1, PD-1 axis. In essence, those patients which have an EMT signal in the stroma were found essentially to see less clinical benefit to nivolumab than those with very little EMT signal surrounding the tumor. And this is shown here in the B and C insets in terms of the panel, where you can split the Kaplan-Meier curve, if you will, by looking at these EMT plus and minus tumors. So again, this is exciting because, again, if we consider that all of this data was generated from the previous data that we had generated off of a single slide, so we're excited in a sense that we're able to generate multiple rounds of data that we can interrogate at different points from that first data set. I wanted to leave off with really the final part really of this discussion, which was really looking at and really looking towards our new precision immuno-oncology panel. Why we're so excited about this panel is the fact that we've really incorporated most of the, I would say, 2018 snapshot of IO as a field. We've incorporated many of the known immunoresistance pathways, including EMT and several others as well as building in tumor intrinsic pathways, many tied to the TCGA molecular subtyping, with the idea that we can essentially give more of a broad-based tool that we can use to really integrate as much biology as we can from a single section to basically to inform retrospective studies. And so in this one example that I'll show that we're getting ready to organize for publication and for posters, so I'm giving you more of a vignette of kind of what we're doing, is looking in the myeloid compartment in non-small lung cancer and showing that in specific cases we can actually detect M2 polarized essentially macrophages or even MDSCs that might be influencing and acting as immunosuppressive influences in the tumor microenvironment to shape or to essentially allow different tumors to immunoscape or to prevent them from being visible on immunological radar. And so what we're excited is really putting a lot of these expanded applications to work. And again, this type of data will work hand in hand with our immunophenotyping application, which will give an idea of what types of immune cells are in the microenvironment as a way to really tie everything together into more of a qualitative big picture. So more to come on this, obviously, in papers and data to come, but we're excited to be able to offer this to the field and certainly look forward to your feedback and have the chance of working with all of you. So I wanted to end the talk here and to really acknowledge the Pierre Santini's group in the Center of Lyon Bard in Lyon, France. We've had a number of really exciting collaborations with him, and we have a lot of exciting studies that we're looking to publish in the months ahead. As well, I want to thank my HDG team, my marketing and R&D team have been working tirelessly with me and collaboratively to be able to pull together this panel, to work together, to really reach out, to understand what is really the critical questions in the field as a whole, and to incorporate this within a new panel for the market. And then lastly, and most importantly, I really want to emphasize, I want to thank the patients and their families for their sacrifices in enabling us to have the types of data that we're looking to build off of to hopefully create even better assays that will hopefully really inform treatments and really bring the most to precision medicine for all of us. So I wanted to thank you all for your time and for attention, and we certainly look forward to the chance to work with all of you in different contexts. So thank you.
And thank you, Mark, for the presentation. It's really informative and interesting, and I think it's a great overview of what HTG's technology can do for people. At this point, I'm going to open the session up for question and answers. You have a questions section in your attendee interface. Please send those questions to us. I do have a few questions that I'll start with. The first question we received a few times was, can the precision immuno-oncology panel be used to measure smaller core needle biopsies that might be derived from a sample cohort with variable characteristics? And the answer is yes. All of our assays have been tested on 416 tissue and specifically core needle biopsies using as small as a few millimeters of tissue. Our recommended input amounts are 5 to 6 millimeters of tissue depending on the assay. And this is consistent with most core needle biopsies, but the assays can use less than that if that is what is available to you to test. The second question that I have we've received is, have you run comparisons of the precision immuno-oncology panel with other technology platforms and other methods? And the answer is yes. As you've noticed, Mark in his presentation talked about some of the comparisons to immunohistochemistry. But all of our panels have been compared to RNA-seq and other competitive gene expression technologies. We have multiple publications in that we can share with you and point that to you if you have further questions on our website. At this point, we'll leave it open for a few more seconds if there's any additional audience questions. If there is not, I will just thank you for spending your time with us this morning or afternoon, depending on where you might be. We really appreciated you attending our webinar. We have one more question just came in. Quick, we'll look at this. The question is, are effector molecules function in relation to their expression level per cell? Are you able to quantitate targets per cell of cell type? I'm not sure I completely understand the question, but the short answer is yes. From our function, as Mark went over in his presentation, we are able to see the presence of different types of cells based upon the relative expression levels of characteristic genes and characteristic receptors. So we are able to certainly quantitate that from a relative standpoint. If it was to do a direct quantitation, certainly using orthogonal methodology would be helpful in that. Hopefully that answers the question. Another question has come in: how much of mRNA or microRNA will be in the blood or plasma normally? That's an excellent question, and it's very difficult to do that and to establish what normal is. What we can tell you or what we generally use is our sample inputs and what we generally derive very good results from. So for our microRNA assays, we are using 15 microliters of either plasma or serum. And we typically see several hundred microRNAs from that input amount. So it's obviously a little bit difficult, and there's quite a bit of loss that can occur trying to extract and quantitate microRNA. That's why our no extraction technology for microRNA is quite useful. For mRNA, we typically use a patch gene or a similar type of preserved blood sample. In that case, we're actually processing a half mil, but actually of that patch gene blood, but actually end up using quite a bit smaller amounts in our assay. So again, we don't typically quantitate that, but we are using the white cell component from a relatively small amount of blood. We'll leave it open for a few further questions. I don't see anything else coming in, so I will sign off again. Thank you so much for spending your time with us. We really appreciate it. And if you have any further questions about HTG, please feel free to go to our website or check further with our representatives. Thank you so much, and have a great morning.